This is episode 107 of the Immunology Podcast, Immune Cell Manipulation with Dr. Rizwan Romee. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Rizwan Romee from the Dana-Farber Cancer Institute on the podcast to talk about his research on genetically manipulating NK T cells. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
we are only 2 months away from IUIS 2025 in Vienna, Austria. Check out the program for the meeting and see what things you can do in Austria by visiting IUIS2025.org.
All right. It's almost summer, Brenda.
I know. Today it's kind of sunny. It's not too bad. I can almost believe summer is coming.
Oh, yeah. It is 80-something Fahrenheit, which is getting close to 30-ish Celsius.
Pretty nice.
Not quite. It's warm. It's a little sweaty.
Yeah. It's borderline comfortable.
Yeah, it's past my comfort zone.
Yeah.
But that's what I'm anticipating. I'm going on my big annual LARPing trip here soon.
Ooh.
Out in the woods, camping in the heat, in armor.
All right. Everything ready? All prepared?
Well, the trailer's over half filled. I still have the rest of it to fill, but I have
to
do that
trailer.
The trailer.
You have a trailer full of stuff.
How else do you bring your yurt and the walls for your camp that you help build every year so that you can siege each other?
Fair enough. That is true. You cannot bring that in a cargo bike like people would do over here.
Yeah, no. The tent that we use is, in addition to the yurt, is over 100 pounds or 50.
That is 50 kilos. Yeah, that sounds pretty heavy for a tent.
It's a canvas tent that's period, so it needs to look good. It's also 20 feet or divided by 3, 8 meters in diameter.
That's pretty nice. I don't know. I'm always amazed at this at your annual pilgrimage to the LARP, the land of LARP. I see you're very committed.
Hey, it started in Germany. This LARP started in Germany.
Yeah, the Germans they know.
LARPing is very serious business in parts of Europe, including like Sweden and Norway and Nordic LARP is its own thing and gets government grants.
No way, really?
Oh, yeah, it's considered a high level of art and gets all these government grants to support it. It's a thing.
Yeah, I mean the Nordics are special people. Let's put it that way.
Yeah, you're just Nordic adjacent.
I'm just Nordic. Yeah, my families want to be Nordics. They always make fun of the poor Estonians. They want to be Nordics.
I have another question for you too. Speaking of interesting things.
Yeah.
Here there's a sandwich that is butter and chocolate sprinkles.
Oh yeah. That's a Dutch thing.
Yeah.
Slagerhach, the slagerhach. It's a breakfast thing. They just put this chocolate sprinkles on top of toast, on top of butter. That's how it is.
Do you eat that?
No.
Have you had it?
I've had it. It's okay. It just feels wrong for me. It's a weird combination.
Is it better or worse than it sounds? Because I'm very doubtful that it sounds like anything other than a heart attack on a piece of bread.
No, it's okay. It's okay. It's not bad. No, I don't like it because it's not my kind of breakfast, but it's fine. It's not. It's just that the chocolate sprinkles on bread, it feels like, I don't know, they just don't go well together, but it's fine. It's a very acceptable breakfast, and I have to say this because they will kick me out of the country otherwise. So I cannot say anything bad about Slagerhach. I already cannot pronounce it. So that's already
it sounds when you chew it, crunch on it in your mouth too.
That must be it. Where it comes from. Yeah. There's a lot of funny Dutch cuisine. I mean, that's usually the small things. That's what's very Dutch. Slagerhach, the croquettes, that's fun in Dutch cuisine. Everything else is kind of potato. But yeah, you should come visit and I'll make sure to bring some over.
It's on the list for the next one of the trips my wife and I want to do without the kids.
It's a bit. I think it's nicer without the kids. Some high European culture. I'll be waiting. Just tell me the word.
Well, first up on my end is, I apologize if I don't get all the first authors because it's not annotated on here, but this is coming out in Nature, published the 4th of June. Concurrent loss of the Y chromosome in cancer and T cells impacts outcomes. First author is Xingyu Chen. Two corresponding authors are Simon R.V. Knott and Dan Theodorescu. And with the dead bodies of pronounced names, we will carry on. So, loss of Y, or loss of the Y chromosome, is already known to be associated with poor tumor prognosis. And this was found to partially be related to the fact that, due to immune evasion from the T cell exhaustion in some tumor sites, in some of the tumor types. They have found that loss of Y in peripheral blood mononuclear sites is the most common somatic alteration in healthy men and has been associated with increased risk of mortality from cancer. So they kind of deep dived what was happening here and trying to understand exactly what was going on. So they did the big bulk of the initial work is they took 29 human types and did bulk and single cell RNA sequencing and looked to come up with a signature for loss of Y. So if you have loss of Y, what genes are changed so you can then predict if this cell has loss of Y or not. So that took a whole bunch of work, and we're just going to say, hey, they got there for the purpose of our discussion. They got there this way, and then looked at mouse models as well. And what they found interesting is that malignant epithelial cells had the highest prevalence of this, but it was also present in tumor stromal cells and in immune cells. And oddly, loss of Y in malignant epithelial cells was predictive of loss of Y in other benign cells, so not a non-cancer cell in the same person. And the loss of Y was correlative if you looked at human and PMBCs. In the tumor of humans versus the PMBCs. If you had one of more, you had one of the other. And so what they found was that loss of Y induced big shifts in the CD4 and CD8 cells, driving immunosuppression, signatures of immunosuppression in their transcriptomes. So if a CD4 or CD8 T cell had loss of Y, you had a suppressive type. And they found that what gets weird is that the independently of each other, how much loss of Y you have in epithelial cells or CD4 cells or CD8 cells all independently predict survival. And if you had all of it going on, it was even worse. And then what they show, and this is where it gets kind of extra zany, is that it seems to propagate. So the final studies that they get into is, okay, so loss of Y is bad. It causes immunosuppression in immune cells. That is bad. It creates more malignant cells and epithelial cells and enhances their signatures of malignancy and bad acting. Okay. But they see that it propagates. And what they get to is, if you have loss of Y in the cancer cells, it causes loss of Y in non-epithelial cells over time. It's what they had in some of the animal models. And that was weird, and they're not sure why. And it then caused them to be exhausted. So they saw that it was correlated in humans. In mice, if they did loss of Y in the tumor cell and then did prolonged activation, that they could get the T cells that didn't have loss of Y, because it wouldn't happen in vitro, but they all became exhausted like they had loss of Y in the human samples. And so something is going on where loss of Y in the cancer affects the immune cell, but then also somehow seems to be associated with loss of Y in the immune cells later on. And they don't exactly fish it out, but it's just an interesting thing that this is a big prognostic marker and seems to be more than just the tumor itself.
Right, but there's no mechanism by which the loss of Y itself affects the outcome, but they could be correlated to something else.
Yeah, so they know loss of Y in cancer cells causes CD4 and CD8 cells to become exhausted. They also see that loss of Y in PMBCs alone is also prognostically bad.
Right, but how do they show that it's just not that individuals that tend to have a loss of Y will also?
They take tumor cells out, put them on in dishes, and then throw regular immune cells at it, and the loss of Y tumors exhaust the T cells.
Okay. Okay. And do like tumors from female cells that don't have Y at all? Is that, would that be a
That's a
control?
control, right? Or male cells that have Y that's there.
Yeah.
So this is a male problem, clearly, right? With loss of
Yes, clearly. Okay.
Y. It's really weird, which I liked. I like weird things.
Interesting. All right, but then there's still, it's just mostly an observation for now. There's not a lot.
Yeah, mechanistically they tie in the exhaustion, but that's about it. I would say it's largely
Yeah.
observational. They can now measure loss of Y, and they see that it's predictive of bad outcomes, which I think that's fundamentally observational.
Yeah. All right. Very interesting. Yeah, I guess it applies to males because we women don't have a Y that we can lose.
So you just have a silenced.
A silence X. Yeah, I wonder whether, well, so many mechanisms. I'm not gonna dwell too much on it, but it's very interesting. All right. So for my first contribution to this discussion today, I have a nice paper from Science Immunology titled "An IL-2 mRNA LNP adjuvant enhances mRNA vaccine-induced CD8 T cell responses." And this is a nice piece of work because kind of you always want vaccines that work better, right? So you always want to find interventional things that you can do for your vaccines to be more effective, and especially I think when we're talking about mRNA vaccines, their original use case was to target cancer mutations and prime the immune system against cancer. And so there are some limitations in our current formulations of vaccines that they're still not completely there yet. And I should probably mention before I continue that the authors, which I have forgotten to mention, are Emily Aunins and Anthony Fan from the labs of Drew Weissman and Christopher Hunter at UPenn. So yeah, makes sense that they're working on mRNA vaccines. And so one of the cytokines that is often associated with a good kind of class 1 response, Th1 response, especially if you're looking for a cancer vaccine, is IL-12. And this is the interleukin 12 is a very well characterized and very well known pro-inflammatory cytokine that is produced basically by myeloid cells, monocytes, macrophages, dendritic cells in response to activation through some kind of pattern recognition or immune activation of these cells. And the targets of this cytokine are, of course, mostly T cells and NK cells. They have the IL-2 receptor, which they can use to sense this cytokine. And in principle, the most important function of IL-12 seems to be the induction of production of the interferon gamma. And interferon gamma, prototypical kind of Th1 cytokine, is very important for orchestrating a cytotoxic response and for CD8 cells. And so it has often, so when we're thinking about ways to enhance an immune response, IL-12 often comes into mind. Can we use IL-12, or is there IL-12 being produced in our vaccination strategy? And what is interesting is that in the case of this mRNA lipoproteins, so the lipid nanoparticles, that actually what they show, it's already been described, but one of the first they show in this publication is actually immunization with this vaccines. They have this mouse models, actually does not result in a lot of IL-12 production by the APCs, which are going to be presenting the antigen that is, the translated antigen to T cells or NK cells or mostly to T cells, and then also activating NK cells, hopefully. And basically, they show that neither the kind of unmodified mRNAs, LNPs, which are not really used because they have very severe kind of side effects, so have been modified to be a little bit less immunogenic. And these modified versions of the RNA, so this methyl pseudo-uridylated mRNA, so this is modified, so it's a bit slightly less immunogenic. If anything, they're better at making, for example, interferon type 1 responses, but not IL-12. And so what they suggest in this paper is, well, what if we if we were putting an mRNA for our target antigen, we can also include an mRNA for IL-12 and get these APCs that are going to be taking up these LNPs to also produce IL-12 as a consequence. So basically, that's what they do. They generate LNPs that have an extra mRNA that is encoding for the two subunits of IL-12, and which are bound together with a little flexible linker. So they are the two parts is just one protein that just binds on itself, and then they have the whole cytokine is there. And basically, they show that if you have this formulation, this combined LNP, you can have production of IL-12 in macrophages and dendritic cells in these mouse models, and that in fact you can have the increased delivery of the IL-12 to potential responder T cells and an increased activation of local APCs. So basically what they show is that although it's interesting because initially they do some experiments where they have like IL-12 receptor knockout mice. And if you have just regular LNPs, they show that there's no difference. Like there's really the IL-12 seems to play no role in the development of CD8 antigen-specific CD8 cells or their expansion. So this really, if you don't, these LNPs really do not depend at all on IL-12 production. But if you artificially add IL-12 to the mix, then this really seems to improve certain important parts of the CD8 response. So you see increased amounts of antigen-specific cells. And what I think was the most interesting part is that they have a kind of a different distribution of phenotypes, and you have a higher population of cells expressing markers such as KLRG1, which often is associated with terminally differentiated T cells, but can also relate to kind of memory homing, so tissue homing for memory cells, for tissue resident memory cells. They also have increased CD69, I think, expression and other markers, and that are associated with tissue residency. And when they look into tissues such as the lung, and I think also some intestine, they also show that they have a bigger population of tissue resident CD8 cells when the mice are inoculated with this mRNA vaccine that has both in this case, they use an OT1 model, so they have OVA plus IL-12. And basically, they show that this combination protects tissue barriers better, or at least you have more infiltration of CD8s on the barriers and barrier tissues. And they also have a model of tumor overexpressing tumor black, I think that's B6 ova, and that again, if you do a vaccination with the LNP with IL-12 and ova transcript, then this results in much better control of the tumor in these mice, better than if you only inoculate them with the LNP with the ova or with only LNP with IL-12 alone. So the combination is really synergistic and keeps the tumors from growing in the mice. So it's nice because you have a much more controlled release of IL-12, because if you put IL-12 systemically, that's very toxic. But then you can have a much more control where you need it, which is in the priming of these T cells. And that really helps generate a population of more effective T cells and what it looks like a better tissue homing of the resulting activated T cells.
Interesting. I always use IL-12 as the devil. So, and is this real
Yeah.
IL-12 or IL-12P40, which is really IL-23?
Um, so in this case, is they have the transcript that they use is codes for both the P40 and P33 subunits of IL-12. So it should be a fully functional IL-12.
All right. I always ask because I was burned by that.
Right. Because people often also measure P40, right? Many of the ELISAs and such, those things they measure P40 subunit.
Yeah. Entire
Yeah.
drugs got to commercial market realizing that they probably should have gone after IL-23 and then did.
Yeah. Yeah. Yeah, that's problematic. But in this case, they have, and also because they're bound by a linker, so these two subunits they cannot be expressed separately.
All right. Well, I'm back to cancer. I'm becoming a cancer immunologist. This is crazy. I'm very confused with myself today. I don't know how I feel about this. So that being said, we're going to talk in immunity, cellular mechanisms underlying beneficial versus detrimental effects of bacterial anti-tumor immunotherapy. Coming out August 12th. First author is Jesse Garcia Castillo. Last author is Michael Dupage. 3 first authors. So in addition to Jesse Garcia Castillo, we have Sebastian Fernandez and Timothy Campbell. So you're familiar with BCG being used as bacterial therapy for some tumors in not in the US, but elsewhere, I think.
I've heard of it, yeah.
Right. So listeria is another one they've looked at because it's intracellular and you can have an attenuated version. And this paper gets very interesting. So they found that if you injected listeria into a tumor, the tumors got bigger. But if you injected listeria IV, followed by listeria into the tumor, or you could just do 2 IVs, one after that, but it wasn't as good. It helped kill the tumor. So that's the main thing here is it's different. But why is it different? That's why you have a paper. But why it's different is if you inject listeria into the tumor alone, it causes intracellular infection of the PMBCs and the neutrophils in particular, and then those neutrophils become MDSCs and are suppressive. So that's the first thing, which is bad. But if you precondition the mouse, in this case, to listeria first, that doesn't happen. And instead, you get tumor killing. And so how do you get tumor killing? Well, they do a bunch of really cool experiments using the OVA system and then either T-cell knockouts and then, RAG knockouts, but then they introduce T-cells from another mouse or what have you to ferret this all out, right? Or these inducible diphtheria toxin to kill cells. But long and short is what they show is that listeria administration generates cytotoxic CD8 cells, anti-listeria cells, and these infiltrate the tumor after you have delivery of the listeria into the tumor, and that they go and kill everything by inducing cancer cell apoptosis. They limit cancer cell proliferation through cytokine secretion, and they enhance tumor antigen cross-presentation because they look at dendritic cells too. But it is not based on these cells, these CD8 positive, detecting, these anti-listeria CD8 cells, seeing listeria in the tumor cell and killing the tumor cell directly. What they're doing is they're going into the tumor microenvironment, seeing that, oh, there's a whole bunch of, infected immune cells here that have listeria, right? And they're taking them all out. And in the process, the tumor gets blasted. And then they're releasing cytokines and signals and porphyrins acquired. So it's causing apoptosis of the tumor from the general war that's happening. And cytokines are being released, which then inhibit production. And then the tumor is getting lysed, and that's causing antigen cross-presentation of neoantigens in the tumor elsewhere. And that's driving the immune response. But it's basically going, oh, there's listeria infected cells here. Let me go kill it. And the tumor is getting, damaged in the process by the immune cells coming in, because they show by ablating the MHC presentation that if the tumor is not presenting Listeria MHC, the immune cells don't care, the effect's still there. But if you get rid of the other immune cells presenting MHC for Listeria, the effect goes away. So it's essentially autoimmune, it's bystander effect on the tumor that is then driving secondary immune responses on the tumor and killing it. But you have to do it by conditioning the immune cells against Listeria with IV, followed by the intraterminal injection. If you do intraterminal first, you just get a bunch of MDSCs, and the tumor gets real big real quick. It's a really cool paper. I mean, I'm surprised it actually didn't make it all the way to Cell instead of just Immunity. But maybe because it doesn't have enough human data these days, I don't know, or it doesn't have transcriptional, a spatial transcriptomics, I don't know. But I thought it had more mechanism and pop to it than some other papers I've seen in higher journals. So shout out to Listeria Power.
Yeah, that's a very cool kind of concept.
Yeah, and they answer all the questions, right? They answered all the mechanism down to how the cells are doing it, with what does antigen required. Like, they don't get into every step of how a T cells affecting cancer, but they're like, look, the cytokines are important, and this is important, and that's important. And like, I read this paper and I was very well satisfied at the end. It felt like a good meal.
The question is, could it ever be done in humans?
Yeah, because this is really attenuated Listeria. It's known to be attenuated. You absolutely could do this in humans.
Yeah.
Give them an IV injection, a week later, inject their tumor. It's not like you can get to any tumor with a needle pretty much.
Yeah,
let's be honest here. There's an old saying in surgery that you can get anywhere with an 18 gauge needle and a firm arm.
Yeah. I wonder if it feels like the poor man's solution, and also there were this really old experiments done. Isn't that the first immunotherapy was basically Pseudomonas, was it? They were injected.
I thought it was BCG is the first one that they first put into tumors.
I think it was something much like, something like Pseudomonas, an actual like heavy bacteria. So half of the people would die, but the ones that didn't, many of them recovered from the tumor.
Sometimes you got a YOLO.
Yeah, I guess. But I think those kind of therapies would be, they're a lot easier. I don't know how difficult it would be. Yeah, the risk, like how much exactly the risk is, but it feels like if we could make things like that work, that would be pretty cool. All right. So, talking about a little bit more sophisticated immunotherapies, my second article for today is titled CREM, that is CREM, is a regulatory checkpoint of CAR and IL-15 signaling in NK cells. First author Hind Rafay, from corresponding author Katayun Rizvani from the Texas MD Anderson Cancer Center in Houston. And this is pretty cool because I think we talk a lot about checkpoint inhibition in T cells, understandably so, and the problem that this baked in inhibitory pathways have in T cell, and using T cells for, serious immunotherapy, and the fact that things like, PD-1 and another kind of T cell inhibitor molecules get expressed as a response to T cell activation, which to some extent makes sense. They have an evolutionary reason to be there, but when it comes to using T cells for therapy, it really gets in the way. So in this paper, they actually identify a similar concept in NK cells, and they show that there is this protein CREM actually is a transcription is the transcription factor cyclic AMP response element modulator, and they identify that this seems to be a crucial regulator of NK cell function. So basically what they do is they start up with car engineered NK cells that you can also put a, for example, CD19 CAR, and the items in this CAR will also activate NK cells. And they look into the effect of stimulation through the CAR and also through another very well-known NK stimulator, which is IL-15. So also IL-15 will activate NK cells. And there is there are certain strategies in which by adding IL-15 expression to your NK construct, you can also improve that. Yes, you can improve viability or survival of these NK cells by having them make their own IL-15 to keep themselves going. So what they look is that they have a mouse model of lymphoma using Raji cells in mice, and they infuse this CAR NK cells into these mice, and they look into transcriptomic the transcriptome of these NK cells using single cell RNA-seq, and they identify this amongst the cells that are differentially expressed in the cells compared from before the infusion to after the infusion. So after they've seen their target and they've been stimulated by their own IL-15, they see this CREM comes as a kind of the top transcription factors that is upregulated. And they kind of zero down on this because of previous research showing that it can be upregulated in response to activation of immune cell activation, and they want to kind of look a little bit closer into that. And they show that indeed is through both the item domain in CDR3 zeta that it's in the CAR, and IL-15 can upregulate the expression of CREM. And basically a lot of other cytokines, IL-2, IL-12, IL-18, they also kind of stimulate CREMs and also other receptors, CD16, and also have items that can also drive CREM upregulation. Basically, it's like this seems to be a molecule that really responds to various ways. Also, NK cells have many ways of activation, so basically they seem to be able to sense different ways in which an NK is being stimulated. And also, when you look into single cell RNA-seq data sets from tumor microenvironments, they also show that CREM is broadly enriched in several immune cells, especially NK cells. And this is from TCGA data, so like human tumor data. So again, they see this relationship between kind of cell activation and CREM expression. And that this is through induced via CAMP, cyclic AMP, and kind of downstream from cyclic AMP sensing, and basically that both of the activation methods that upregulate CREM are affecting this pathway. So what I think the interesting part is that they see, we see that there's this molecule is upregulated upon several ways of activation, and in NK cells they seem to be really reproducibly upregulated. What happens if we remove it? And I think this is kind of when it gets interesting, because they show that if they knock out CREM in the NK CAR cells, actually that makes the cells work a lot better in vitro. They have this, they do some co-cultures, and basically they see that the cytotoxicity improves a lot, and also upon re-challenge. They have these nice kind of little videos. So removing this molecule actually improves NK function. And this really is the kind of, if they, and this really only gives a benefit if the NK cells are being activated. So it's really showing that the CREM is kind of blocking some of this activation signal going further, because they have, for example, these item mutations in which the signaling is abrogated, and then there's no effect of knocking out CREM. And when it comes to using, mouse models and kind of really disease models, they have, again, this aggressive lymphoma model, and they show that if they have CRM knockout cells, they have better survival and better proliferation and control of the Raji cells, of the lymphoma cells. And they also have some results on a cancer model, breast cancer. And again, if they treat them, they're CD70 positive breast cancer, so they have CD70 CARs. And again, knockout of CRM reduces tumor burden and improves the proliferation and tissue infiltration. So they do some, epigenomic analysis, ATEC-seq, and they show, kind of the impact of this CRM knockout, and they show kind of some mechanism by which this activation is affected by CRM. And basically, they suggest that if you can by knocking out CRM, you can enhance the cytotoxicity of CAR NK cells, and this is only is you done in conjunction with signaling through an ITAM and for example IL-15, addition of IL-15 to the construct to really get the NK cells very well activated.
Interesting. So there's a joke here about the creme de la creme.
That is true. The creme de la creme. But then it's a good thing. In this case, a bad thing.
Yeah, but it's
you don't want
creme de la creme of NK cells. Is it the cream? I don't know.
Yeah. Keep working on that. We'll work something out. So making the NK cells. I don't think a lot about NK cell therapy sometimes, but I think it's I should.
You should be, because we're about to talk to Dr. Rizwan Romee at Dana-Farber Institute in just a moment about NK cell therapy, Brenda. But before we get to that, do you do immunotherapy research, Brenda? STEMCELL Technologies offers products and protocols for immunotherapy research, including T-cell isolation, activation, and expansion reagents. Use EasySep T-cell isolation kits to isolate highly purified T-cells in as little as 8 minutes. Follow up with Immunoccult reagents designed for human T-cell activation and expansion. Learn more about STEMCELL's optimized protocols and reagents for immunotherapy research at www.stemcell.com/T-cell-therapy.
So now for the second part of our show, for our guest, we are really happy to have with us Dr. Rizwan Romee. He is an oncologist at Dana-Farber Cancer Institute and associate professor at Harvard Medical School. And the Romee Lab has really cool research manipulating NK cells for therapy, understanding, how to use innate cells for genetically engineered therapies. And that is particularly the part I think I find most interesting, but that's not all there is. So I'm hoping that we will get to all the research in this interview. So Dr. Romee, thank you so much for joining the Immunology Podcast, and welcome.
Well, thank you, Brenda and Jason, for hosting me. I look forward to having this chat with you guys.
Thank you for being here. All right. So I guess the easy way to describe some of what you do, and tell me if I'm wrong, is engineered NK cells. Essentially, some people call CAR NK, but not necessarily CAR. Why use NK and use engineered NK cells? What's the advantage you were getting versus your classic CAR T at this point? Want to call it class?
For years, people have studied T cells, engineering both CAR as well as TCR and other genetic manipulations, and have made decent progress in recent years, and there are several products approved. However, there are two issues with the CAR T cells and other T cell based approaches. One is that the safety. So every time people have used T cell based approaches, they have seen what is called cytokine release syndrome, and sometimes it can be very severe. And then also things like neurotoxicity, and more recently, even though rare, people have seen lymphoma formation from the T cell themselves, as well as other rarer complications like HLH, which is basically bone marrow failure and this pro-inflammatory or this prolonged inflammatory state. None of those things happen with NK cells. And in addition to the safety, obviously, we think that NK cells can be more potent in some ways. They not only, if you put a CAR on them or a TCR on them, they not only target through the TCR or the CAR, but they have this intrinsic propensity to target tumor cells, which gives them a huge advantage. So you are basically dealing with the cell type which is born to kill. That's part of the reason why they're called natural killer cells. They're born to kill. They're trigger happy. They're kind of first line of defense against infections and tumors in our bodies. They are ready to go. They don't need to be primed versus T cells. They need to be primed. They need to be trained versus NK cell ready to go. As I said, trigger happy cells, and yet they don't cause toxicity. So that makes them pretty cool, in my opinion. And that's part of the reason why I think there's plenty of room for trying other cell types, including natural killer cells, as an armamentarium, as part of the armamentarium against different cancers or different malignancies.
So let's just take a step back and go to a little bit of NK biology 101. So for our listeners to get their right mindset. So unlike, as you mentioned, T cells, T cells have a T cell receptor and they recognize. So when they recognize the receptor under the right circumstances, boom, they get activated and hopefully they do their job. But NK cells don't work like that, right? So you have this kind of balance between inhibitory signals, activation signals, and it is kind of the net the net value of these two that will dictate the function of an NK. So why don't you maybe help our listeners understand which are these different signals and how do you modulate them or how do you exploit them for therapeutic purposes?
Right, this is a great question and also a nice description of kind of overview how NK cells decide to kill or not to kill. As you mentioned that, as with actually any immune cell type, NK cells have a bunch of receptors on their outer membrane. So you have the inhibitory, you have the activating, you also have, homing receptors, you also have receptors which respond to other cues in the tumor microenvironment, also, to other cues in the body in general. But if you focus on the receptors which respond to the tumor targets, and also some of them bind to normal healthy cells, there's, as you said, there's a balance that, yes, NK cells are trigger happy, yet that trigger is held back unless the NK cell makes it absolutely certain that the target is a bad one. Either it is infected or has undergone some stress or is malignant, has undergone transformation. And in general, there's this class of receptors on the mature NK cells called KIRs, the killer cell immunoglobulin-like receptors, which bind to the HLA molecules. As you know, most of the nucleated cells in our bodies have class 1 HLA molecules, class A, HLA A, B, and C. And many of those, particularly in the C family, C1 and then C2s, and some BW4s and HLA-As bind to the inhibitory KIRs expressed by NK cells. So every cell, when it interacts with NK cells, as long as it's healthy and maintains the HLA class 1 molecule expression, will try to shut down an NK cell. However, NK cells also have a group of activating receptors. For example, NKG2D, which binds to MICA, MICB, and ULBP proteins. And those proteins normally are not expressed by normal healthy cells. So you can imagine a normal healthy cell doing its business, and NK cell checks it out. Hey, buddy, are you okay? And it tells the NK cell, well, I have the class 1 molecules, and I don't have those stress ligands, or I don't have those activating cues. So NK cell, okay, I'll leave you alone, and moves on to the next cell to assess its, stress state or malignant state. But you can imagine the same cell, once is infected or has been exposed to radiation or is really old, sometimes aging, the senescence can also trigger NK cells. They will either lose the class 1 expression, like lower the expression, which often cancer cells, for example, use to escape T cell attack. By the same token, that leads to less of inhibition of the NK cells, which is checking status. And in parallel, oftentimes the malignant cells or stress cells or cells which have gone radiation or infected, they have increased the expression of those stress ligands, classic being MICA and MICB. And certainly there's this balance where normal state, homeostatic state, this was in favor of net inhibition, but now certainly this malignant cell, this tumor cell, has lost or lowered the expression of class 1, which would have normally inhibited an NK cell, but there's lack of inhibition. And yet there's increase in the expression of proteins which actually activate an NK cell. So the balance is shifted or tilted towards activation and the trigger gets pulled. And once the trigger gets pulled, several things happen. NK cells will dump perforin and granzyme, and that leading to the hole formation in the membrane. And granzyme goes in, it triggers the apoptotic machinery, and cell commits suicide. The tumor cell commits, or infected cell commits suicide. Then also the activated state, they secrete several other things. For example, interferon gamma is secreted, TNF alpha is secreted, which can also modulate the tumor microenvironment, actually make it more pro-inflammatory state. They also release chemokines like CCL5, which will bring in other cells, including T cells and macrophage into the tumor. So it kind of helps trigger this immune attack once it determines that the cell which is interacting with is not healthy. So that's how it kind of an NK cell unleashes its kind of bad thing, not bad for the body, but bad for the target cells, and tells the body, hey, there's a problem here. We need to bring in more soldiers, other types, not just the NK cells.
So it sounds like because NK cells really rely on this specific positive signal, you're going to have less chance for that immune activation that can go on with T cells and autoimmunity and other stuff. Are NK cells relatively resistant to an autoimmune situation or other large cytokine storms? Because they're not, I know everyone throws out cytokines, but they're not, are they spewing out enough to pull everyone else in? Or they're mostly, hey, buddy, you good or not? You're not good, you're dead.
That's, Jason, the right way to describe it. They are like that. So there's less chance of an, autoimmune attack and less cytokine storm. Actually, my lab, in some unpublished data, we have compared head-to-head same NK cell, meaning same patient or same donor NK cell, same donor T cell, put the same CAR on them, and then put the same target, and then let's look what they spew out. And there's huge differences. So the CAR NK cells in this case produce way less quantities of things like IL-2, IL-6, and things like that, which are associated with cytokine storm. So yes, the stuff they produce, the way they produce, they are this balance of inhibition versus activation that makes NK cell-based therapies safer.
Yes, that's pretty interesting because, yeah, indeed, the cytokine release syndrome is quite an impediment sometimes and has been very problematic in many T cell therapies. So I see that you are, of course, an oncologist, and for what I understand, you are contributing or with clinical trials. So you are, really there, interacting with the patients. And so, what has been your experience? Because I hear a lot about NK therapies, the potential that it has, because then you could do them allogeneic, so you can have one set of really good NK cells, and you can treat many patients with that. So, how has been your experience so far with NK cells in the clinic that you would like to share with us?
Yeah, no, NK cells, as you just mentioned, and also the safety, thing. In that context, NK cells have a huge potential and are attractive, especially if you are going to go with the allogenic route. They are even safer because they don't. When you take a T cell allogenic, it can cause what is called graft versus host disease, because some of the TCRs can recognize the host normal healthy cells, through the HLA mismatch. But the NK cells don't have TCR, so you don't need to worry about graft versus host disease. That's why part of the reason why NK cells are very attractive for developing allogenic products. However, so far, we haven't seen a single NK cell product getting approved. And that comes to the experience, my own experience, and also the field. The biggest challenge I see for NK cells, for the efficacy, yes, there have been multiple trials done, including our own center here at Dana-Farber. I lead some of the trials. I'm the IND holder on some of those trials. But what is the biggest challenge with NK cells? Their half-life. So NK cells don't last long. And I firmly believe that applies to almost any cell type. Unless they last at least a month or two or more, they may work, but the durability will be a problem, or they may not work at all. And this is what we are seeing in, for example, CAR macrophage world. So some of the recent clinical trial data from CAR macrophages has not been that great because of the fact, again, the in vivo the inside the body, the half-life of those cells, including NK cells, is short. So that's been kind of the biggest challenge to increase the efficacy or to see the efficacy potential of NK cell products. And to be honest, anything beyond T cells. So T cells, on the other hand, we know some T cells can live forever. If you have memory formation, they can really live forever. And there's a paper from Carl Jones group where they found this first CLL patient, or one of the first patients who was treated 10 years back, or more than 10 years back, still had those CAR T cells in small numbers lingering in the body. So that speaks the power of persistence and power of living long inside the body to maintain remission. So I think that's been one of the challenges, and that's part of the approach. Well, that's part of the challenge we are taking in my group, in my lab, where we are really trying to figure out, yes, CAR NK cells or our NK cells, engineer NK cells work, but how do you increase their persistence in the body? And that's one of the things which I think has to be overcome in a significant way before I see NK cells getting approved and showing their real potential in clinic.
So is there engineering that lets you do that?
Yeah, so there are several approaches. My lab is taking the cytokine engineering. As you know, cytokines are quite powerful molecules, and what we have seen is that when you activate NK cells, regular conventional NK cells with certain cytokines, particularly IL-12 and 18, it can induce this memory-like phenotype, and those cells can live longer. So that's one approach. But I still think that's not enough. More recently, we have tried different cytokines engineered into the genome along with the CAR into NK cells, and found that IL-12 in particular, people have used, obviously, MD Anderson group and several other groups have used IL-15 secreting CAR NK cells, and shown some potential promising efficacy. But my group, when we compared different cytokines, we found that IL-12 single-handedly beat IL-15 CAR NK cells, meaning these are the same donor NK cells which produce IL-15 versus our IL-12. And we see those cells persisting much better in the mouse models, and also trafficking better to the tumor microenvironment. So that's one approach. That's cytokine engineering, and people are trying different cytokines. My group is betting on IL-12. Now, historically, IL-12 has been very it's a sensitive issue because people have tried IL-12 in CAR T cells and T cell therapies, and has been found to be very toxic. But our data shows that IL-12 secreted by NK cells is way safer than IL-12 secreted by T cells. Again, it comes back to the secretome, right? What are the T cells secreting when they have IL-12 in? What are the NK cells secreting when they have IL-12 knocked in? And the secretome profiles look very, very different. There's an hopefully upcoming paper, it's under review currently, where we're touching some of these issues.
So is this IL-12 or IL-12 that's IL-23?
No, it's IL-12. So I know it's a big family. People are trying, different IL-12 family members, but we have engineered IL-12 a little bit. It can maintains its efficacy, but it tends to get trapped in the tumor matrix. That prevents systemic exposure, making it safer, and yet allowing not only cis activation of the NK cell CARs, but more importantly, allowing tumor microenvironment modulation, where it affects T cells, NK cells, and other, including macrophages, which are already present in the tumor microenvironment. So this TME modulation is something I'm betting big on. I think as a field, we need to see that more, especially in solid tumor settings, where one cell type is unlikely to cure anyone.
Just to clarify, why would IL-12 and IL-23 be confused with each other? So what is the background behind that question?
They share a subunit.
Yeah.
And it was a bunch of confusion for decades, or for like a decade. They even made drugs that are anti-IL-12 that are on the market, but made anti-IL-23 drugs afterwards when they realized all the mechanism of action was from the IL-23 part, because they made an antibody and they made it, monoclonal to the shared P40 subunit.
Right.
IL-12 P40 is used in IL-12 23 as part of IL-23.
So so adding to this comment from Jason, I'm glad you brought it up. So Brenda, IL-12 family includes 4 members. You have IL-12, IL-23, IL-27, and IL-35, and they share different subunits, so the receptor, and as well as the subunits of the molecule, like P35 and P40 and the combo. But so they share some of the properties, and depending on what receptor they bind and what is composed, so you can have totally different. As with any cytokine, the context matters, and obviously these have different downstream pathways. So there's this drug which was developed against I believe it's P40, but which is shared between IL-23 and IL-12. And it's called Stelara. That's the name, the compound, also called ustekinumab. And ustekinumab was, I hope I'm pronouncing it right, it was developed for inflammatory bowel disease. And it's really good at neutralizing both IL-12 and IL-23. As a matter of fact, one of the things we are doing, and we have shown it's again in this paper, hopefully upcoming soon, where we showed that when the CAR NK cells secrete IL-12, if you use this tumor matrix binders, it gets trapped in the tumor matrix. There's minimal exposure to the rest of the body. However, to make it more strict or make it more stringent, like in terms of preventing exposure in the body, we injected ustekinumab and showed that we can neutralize IL-12 to a large extent. So that's another safety mechanism which we can use, because Stelara or the ustekinumab is already approved for inflammatory bowel disease.
And interestingly, the IL-23 version, which is gazelkamab or Tremfya, same company, makes both, outperforms the Stelara.
That's interesting.
For IBD.
For IBD and psoriasis.
Interesting.
For autoimmune diseases. So there may be an anti-autoimmune effect of a little bit of IL-12. Want that you don't have this, dual effect issue when you have a pure IL-23 blocker. But it's right. An interesting quirk of the discovery was moved on so fast that pharma made a drug before they had a oops.
But it's interesting that if you look at these molecules like IL-12, 23, 27, and 35, starting from 23 on one end, that's the most pro-inflammatory molecule. It actually leads STAT3 and STAT4 activation downstream, versus IL-12 is only STAT4. And then as you move towards higher up, that's how I remember, as you move from 23 to 12, both pro-inflammatory. Then 27 is kind of in between, still pro-inflammatory. But the 35, which is also a member, is completely, or not complete, but it's predominantly inhibitory molecule. So it's interesting, this spectrum. And I guess the reason this new drug works better in IBD is because IL-23 is more pro-inflammatory than IL-12. That's just my guess, not being an expert in autoimmune disorders.
That makes sense. But it says really, I don't want to linger too long in this, but the idea is that, if they share subunits, like, usually when people discover this, I don't know, now you have very powerful, proteomics, but I guess that if you look at papers from 20 years ago when they're discovering these cytokines, and you have these Western blots in which, and then you couldn't tell the difference, right? If the two subunits are the same and you miss the third, and you're like, I see exactly the same. I see these two things, I get the same pattern in a Western blot, so I guess they must be the same molecule, or I guess I cannot tell the difference. So it's not that crazy. Sometimes I think we overestimate how much we can do now, but often this first cytokines, the way they were described, is pretty artisanal, the kind of experiments that were done. So I wouldn't take it too, I guess, it's not that strange that it was.
Yeah, I mean, but that speaks volumes about what is happening now in protein biology in general. So every time we come with a new sequence, or we want to try some new sequence, before we even try the biologic activity, right? So my lab works on a lot of protein engineering, particularly creating this fusion product, and we almost every single time run the alpha fold and to see what kind of and then we can actually nicely predict or not whether it binds to a given receptor. So it's interesting how the protein biology, protein engineering has gotten very simple. Still, it needs a lot of work, but at least it's more accurate and simpler. At least you have the tools easily available on any desktop, where you can actually see, okay, can this particular protein which you are trying to develop or discover bind to a given just based on the sequence. It's kind of
Yeah.
interesting how people back in the day would spend their entire careers on X-ray crystallography, right, to predict the full. And now with the AlphaFold and technologies like that, you can at least, to a large extent, it may not be 100% accurate, but it's getting better and better. So, machine learning and prediction tools are getting really, really good at predicting the exact three-dimensional structure, which is amazing.
Yeah, no, it's really interesting. So one more question before we probably switch to the non-science part. Made some really cool E. coli.
Oh, yeah.
Non-pathogenic E. coli. Why are you shoving an IL-18 mutant into E. coli?
I'm so glad you asked this question.
Is this a way to get IL-18 in, or is the E. coli doing something beyond just being a IL-18 vector?
So the short answer is we are using E. coli as a vehicle to deliver biologics. And even though the recent paper, which we published in Nature Biotech, delivered this mutant of IL-18, the decoy receptor, resistant IL-18 to solid tumor setting, but we have gone beyond that, and we are developing all sorts of biologics using our bacterial platform. So E. coli is a gram-negative bacteria. There are several hundred strains. Turns out some of them are very non-pathogenic, meaning we have them in our gut, and that's the one we took it, and you can safely inject. They're not toxin. They don't have any toxin islands, and they're very pan-sensitive. But the interesting feature about these strains is that they're facultative anaerobes. They allow low oxygen environment. They also allow low pH environment. And tumors, many solid tumors have both of them. They are high lactate, low oxygen, and the vasculature is leaky. And the tumor microenvironment is immune suppressed. So we found in this mouse model, in the paper, that we inject intravenously this bacteria, there's 10,000-fold enrichment in the tumor microenvironment. We use the MC38, which is a colon cancer model. And the reason we think is that, again, they allow to proliferate in high lactate, low oxygen, and leaky and the tumor suppressed microenvironment. So then we said, why can't we take advantage of these cells? And they allow to go to these areas which CAR T cells and CAR NK cells have a problem in going. Why not then deliver biologics? And in this particular paper, which we published recently, we delivered IL-18. We actually tried IL-15, IL-21, IL-18, but IL-18 showed the most efficacy. So we are working closely to take into clinic because, as I mentioned, these strains of bacteria are non-pathogenic, yet they are tumor homing. It's not actually homing. We don't think they actually seek the tumors. It's just that those things which I just mentioned, high lactate or low pH, low oxygen, highly immune suppressant, it just favors their growth there. And so it gives us a nice Trojan horse strategy where we can deliver biologics into the tumor microenvironment and potentially even bring other immune cells and toxins and whatnot to help fight the cancer. So I'm pretty excited about this approach we are taking.
So I guess my last follow-up for this, being in pharma, working in the microbiome space, is the FDA concerned about the fact that it's a genetically engineered E. coli? Have you built in a kill switch so you have to feed it like a custom sugar or something? Because I know that it's one thing to give commensals. It's another thing to give something genetically engineered that could proliferate endlessly and then be pooped out and then go into the microbiome ecosphere. And that's a huge concern of theirs.
That is. But to address this, so what we did was we deliberately put a plasmid-based system. We didn't do the actual genome. So they are not permanently genetically engineered. So we put these plasmids, which have the IL-18 and other biologics and other inducible promoters. So if you have bacteria, we don't have that induction system. They won't have the expression of a given biologic, and they last only 10, 12 days. And we have done extensive studies in this mouse model, but after 10, 12 days, they lose the payload, and also they themselves get killed. And then also in terms of kill switch, yes, we are actually developing a CRISPR-based kill switch, which we haven't published yet. It's in collaboration with my friend and a colleague at Ann Arbor, who is also co-corresponding author in this recent paper, Professor Jae Lee. We are developing CRISPR kill switch to kind of make it even more safe or make it safer. But then also this particular strain is pan-sensitive. It has no antibody resistance to most of the commonly used antibodies. So there we have a kill switch of using simple as pan or strep or any other or doxycycline or any antibody just dies. So yes, we are not creating permanent strains which can spread to other people because they are not this editing doesn't involve the bacterial genome. It involves only the plasmids, and plasmids don't last. And also these bacteria don't have the recombination apparatus, so they can't spread that plasmid to the neighboring. So that makes it even safer. So all of those things. But yes, the last thing you want is that yes, this is cool for cancer cure, but then we don't want to create a new strain which spreads the word and spreads our biologics, IL-18, because that could have a disastrous consequences. So I'm fully aware of that.
That's pretty cool, I have to say. That's really,
Thank you.
and it looks like you've seen it through our angles. I think as Jason makes the point, safety is very important.
Absolutely.
But I really like we've also covered in our show and looking at papers is about using these bacteria to deliver therapeutics. I think it's a really cool approach.
I think the comment I have on Brenda is that, for a long, long time, bacteria were there was a lot of attraction towards using bacteria. I mean, look back like several hundred years of Louis Pasteur,
Yeah.
right? I mean, that's the whole idea. And even coli's toxin that was actually strep-based injection directly into tumors in 1890s. And if you look at early 1900s, he actually published, Professor Cooley did publish several reports of actually tumor shrinking, but then also it was at the time, we had very bare minimum knowledge about immunology and also how the bacteria work. So unfortunately, a lot of the, we had obvious infections. We are causing infections. But now with the synthetic biology tools, with the modern, recombination technologies, synthetic biology in general, we have become smarter, I think, and we can harness their power in a much, much better, safer way and also more efficacious. There's this new resurgence, if you will, of interest in using bacterial-based therapies.
Yeah.
Which is exciting.
Yeah, absolutely.
Thank you.
Absolutely. Well, on that high note, let's move to our last question of the day, in which we try to get to know you a bit better for the benefit of our audience. And this is a question I really like to ask as well. What is a hobby that you've always wanted to pursue but haven't had the chance to? Maybe when you retire, you'll get to it, or something that is a hobby that or maybe you will make the time, but what's the relationship between you and hobbies?
Yeah, no, I love this question. So when I'm not doing science, I love to be in nature. And over the years, I have fallen in love with bird and wildlife photography. So I spend a lot of time going to wildlife reserves. But so that's my hobby. And also I want to make a comment. I don't ever plan to retire because I love science too much. I can't imagine my life without science. So no matter what, it's hard to take me away from science. Now, one hobby I really have always wanted, but haven't done is scuba diving. And the reason for that is I love marine biology. If you ask me what's your favorite animal, it's going to be orcas or octopuses, or octopi, I guess, because they're so cool. There's so much novel biology there. Like, how cool is that? You have the hemocyanin, rather hemoglobin in octopus, right? And they're very super intelligent. Similarly, orcas, they are super intelligent. There's language, they live in pods, they communicate with each other. Super, super intelligent animals, and so much to be explored. So I would love to pursue marine biology as a hobby and do scuba diving, but I haven't gotten a chance to do it yet, but can't wait.
That sounds fun. Also, I might say, the orcas, aka the killer whales, like the killer cells. So I think it's meant to be. There's a connection there. If you ask
there you go. I love that connection. But being able to spend time with these marine animals, like I love fish, but I particularly like the orcas and octopi.
Yeah.
It's like getting close to them, touching them, or not even touching, just being close in the environment, right? It's just amazing.
It can be a fun investment, I will say. Like a lot of my friends have done the certification, and you have to take some classes, but you feel the progress. You get there. You can do it over the course of a vacation pretty easily.
Or rent a sub, right? The people go down. Although we had a crash, so that's a little
Yeah,
scary.
you don't want to go to those
No.
ones. Or you can go to those places where you can like this harbors where the whales go and they just hang out. And if you're lucky, you're just like paddleboarding and there's
Yeah.
whales around you.
Yeah.
And then if you're unlucky, they might eat you up and spit you out. But you
Yes, I love that, right? The guy who was eaten like was spit by. Yeah, no, not in orca's mouth though. That'll be
yeah,
disaster.
that's the mouth you don't want to get in. That's for sure. That sounds
Baleen
fun.
whales, they can't swallow. That's fine.
Yeah, exactly.
But not the orcas. But
Oh,
then
well.
marine biology, yeah.
All right. So I think with that, we can conclude our conversation for today. It was really, really interesting to hear. I think the engineering of NKs and bacteria, I think is just absolutely fascinating. So thank you so much for joining us. We had Dr. Rizwan Romee from Dana-Farber Cancer Institute and Harvard Medical School talking to us at the Immunology Podcast. So thank you so much. And well, hopefully we'll see you in the future.
Thanks, Brenda and Jason. Thanks for hosting me. It was fun chatting. Bye.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.